Guys, so I'm going to walk you through our DNA replication diagram. So the document is linked to Google Classroom, so you can either print it and fill it in as we go, or you can simply draw it out if you don't have a printer. Shouldn't take you too long there. So what we have here is a double-stranded helix of DNA that is getting split apart, right? So the first thing to look at whenever you're looking at a replication diagram is the directionality of the DNA strands. So what that means is that DNA is always read in a particular direction, and the way that we know what direction it's being read in is we actually give a number to the direction. And so in order to figure out which is the 5' end and which is the 3' end, we have to look at the sugar in DNA, which is deoxyribose. Deoxyribose is a pentose sugar, meaning that it contains 5 carbons. So what I did was I went through and I already numbered them for you. So 1, 2, 3, 4, 5. So whatever the last molecule is will help determine the end. So this part right here, we can't see the individual nucleotides because this is a simplified picture here, but if we were to see the molecules, this being the 5' end, that would mean that the phosphate here would be coming off the 5th carbon. This would be the 3' end because if we could see the actual molecular structure, we would see that there is an open bond on the 3rd carbon. So remember that DNA strands run anti-parallel. So this top strand here goes 5 to 3, but the bottom strand goes 3 to 5. So they run opposite but parallel. So that's just a little intro on the 5' to 3'. Next, we're going to look at actually how DNA replication takes place. So the very first thing that has to happen is the DNA strand must be separated. And in this diagram, the enzyme that's doing that is letter B. Okay, and the enzyme responsible for that is going to be DNA helicase, okay? Think helicase works on the helix. And so it opens up these 2 strands for new enzymes to begin work. So we'll look at this top strand here first. This was a simpler one. This is called the leading strand. Okay, so J here. J here is referring to the leading strand, and this one gets synthesized or made first. This one gets synthesized continuously in the 5' to 3' direction. All right, so that will take place continuously, running anti-parallel to the template strand, so it will go in the 5' to 3' direction. So what this little red piece here, okay, this is actually an RNA primer sequence. And that pretty much tells the enzymes where to begin replication, right? Because they're just kind of floating around and they don't exactly know where to start. So this is actually a sequence of RNA that is placed, and it's simply there to tell the enzyme, okay, here's where you're going to start. So K is actually the enzyme responsible for this, and it's called primase. So the enzyme primase lays down an RNA primer sequence which tells G, which is DNA polymerase, where to begin synthesizing that leading strand. So once this strand is all made, then the cell begins to work on the lagging strand. And again, the K down here stands for the same enzyme as K up here, which is primase. So the lagging strand gets synthesized or made in chunks. So again, it will be made in the 5' to 3' direction. And it's laid down in several fragments. Okay, so again, all of these H's right here, these are the primer sequences. I'll just put primer, which are laid down by the enzyme primase, which is what K is standing for. And they get laid down in fragments. So each one of these fragments is called an Okazaki fragment, just named by the scientist who discovered them. His last name was Okazaki. And then last but not least, once these individual fragments have been placed down, DNA polymerase will actually come back and remove all of the RNA sequences because you really don't need those. It takes those out and replaces them with replaces them with DNA nucleotides. And last but not least, something that's not shown here is our enzyme ligase, which links the fragments together. So really quickly, just to run through this with you from the very beginning, helicase unzips the DNA. An RNA primer sequence is laid down by primase, which tells DNA polymerase to start here. The leading strand is synthesized in the 5' to 3' direction. Once this one's all done, more RNA primer sequences are laid down on the lagging strand, but they are done in chunks. DNA polymerase will then come and start to lay down new nucleotides on the lagging strand in these fragments. Once all the fragments have been placed, DNA polymerase will go back and remove all of the red bits, which are the RNA sequences, replace them with DNA sequences, and ligase will link all of the fragments together. All right, so that's pretty much DNA replication if you were to look at it on this diagram. I recommend watching YouTube videos with this happening in a continuous loop just so you could visualize it a little bit more.